In interphase of meiosis, chromosomes replicate during S phase but remain uncondensed. The centrosome replicates, forming two centrosomes. In prophase 1, chromosomes are now fully condensed. In crossing over, the DNA molecules in non-sister chromatids break at corresponding places and then rejoin to others' DNA.
Wait!
Has one or more chiasmata, crisscrossed region where crossing over has now occurred. These hold the homologues together until anaphase 1. The movements of centrosomes, formation of spindle microtubules, breakdown of the nuclear envelope, and dispersal of the nucleoli occurs in mitosis. In late prophase 1, the kinetochores of each homologue attach to microtubules from one pole or the other. The homologues pairs then move toward the metaphase plate. In metaphase 1, the pairs of homologous chromosomes in the form of tetrads are now arranged on the metaphase plate, with one chromosome of each pair facing each pole. Both chromatids of a homologue are attached to kinetochore microtubules from one pole. Those of the other homologue are attached to the microtubules of the opposite pole.
And then what happens? There you go.
In anaphase 1, the chromosomes move toward the poles, guided by the spindle apparatus. Sister chromatids remain attached at the centromere and move as a single unit toward the pole. Homologous chromosomes, each composed of two sister chromatids, move towards opposite poles. In anaphase 1, the chromosomes move toward the poles, guided by the spindle apparatus. Sister chromatids remain attached at the centromere and move as a single unit toward the pole. Homologous chromosomes, each composed of two sister chromatids, move toward opposite poles. Stop. In telophase 1 and cytokinesis, at the beginning of telophase 1, each half of the cell has a complete haploid set of chromosomes, but each chromosome is still composed of two sister chromatids. Cytokinesis usually occurs simultaneously with telophase 1, forming two haploid daughter cells. In animal cells, a cleavage furrow forms. In prophase 2, a spindle apparatus forms. In late prophase 2, chromosomes, each still composed of two chromatids, move towards the metaphase plate. In metaphase 2, the chromosomes are now positioned on the metaphase plate as in mitosis. Because of crossing over in meiosis 1, the two sister chromatids of each chromosome are now not genetically identical. The kinetochores of each chromatids are attached to the microtubules extending from opposite poles. In anaphase 2, the centromeres of each chromosome finally separate and the sister chromatids come apart. The sister chromatids of each chromosome now move as two individual chromosomes toward opposite poles. Go. In telophase 2, nuclei form. The chromosomes begin to decondense and cytokinesis occurs. The meiotic division of one parent cell produces 4 daughter cells, each with a haploid set of unreplicated chromosomes. Each of the 4 daughter cells are now genetically distinct from the other daughter cells and from the parent cell. Nuclear fragments have now reappeared, ready to become full-fledged nuclei. The 4 daughter cells now live happily ever after.